self-assemblies to tissue engineering.
Thank you very much for the kind introduction. So, ladies and gentlemen, good morning. My name is Dr.B.Nottelet, as just was mentioned, and I'm from the Artificial Biopolymers group. So, the main topic I will try to develop today is about the preparation of novel degradable synthetic polymers. So, I won't talk about natural polymers, but only synthetic ones. And our group is working on this topic for about 30 years now. And we are mainly involved in the use of degradable polyesters. I will try to show you that thanks to the synthesis of new polyesters, it's possible to control the drug of delivery with some examples in terms of self-assembling structures and also tissue engineering. But before starting, I'd like to give you just a quick overview of the development of polymers as biomaterials. Most of you may know this, but it started more or less in the 70s with first generations of polymers that had more or less to mimic the properties, mechanical properties of the tissues to, for example, function repair applications like cardiac valves or lenses. In a second step, the next generation was made to be integrated into the tissues, so degradable polymers were necessary for wound repair applications like the famous osteoconductive applications. And finally, we are now at the third generation of polymers where we want to have polymers that can induce cell response at a molecular level, but also have the good mechanical properties. And that's what is used in tissue engineering applications like skin scaffolds. We had an example this morning. And that can be obtained through many techniques like surface modification, drug encapsulation, and vectorization also. Beside all these applications, what is interesting with polymers is, of course, that you can play with them in terms of shapes and phase in which you can find them. You can have, for example, the aqueous solutions of micelles, aggregates, or polymerosomes. Then the solid nanoparticles or microparticles, hydrogels, elastomers, scaffolds for tissue engineering, or even purely solid matrices. So that's something which is quite broad and interesting in terms of engineering of the materials. In our group, we are, as I said, dedicated to preparation of degradable polymers, and the group was founded by Michel Vert, who started about 30 years ago to work with metabolites in order to obtain monomers. And those monomers, which are new lactones in most cases, can be polymerized to generate degradable and biocompatible polymers. And the main aim is to have, through the degradation of the polymers, the release of non-toxic byproducts, which might be also metabolites. Those metabolites can also be used for targeting or bioactivity. We had an example this morning of peptide sequences with the RGD. All the folic acid or mannose targeting moieties can be also used, etc., etc. So that's somewhere we are trying to work. And to do so, just to give you an update of the group, since 5 years, the group is headed by Professor Jean Coudan, and we are an interdisciplinary group with half of the people involved in chemistry, and the rest involved half in the characterization of the materials, and we've got also a pharmacist and a medical doctor for biomedical evaluation of the systems. So, with this, I'd like now to start with the scientific part of my talk. And what I'd like to show you today is that through simple chemistry, compared to, for example, organic chemistry for medicines, we can, even with this simple chemistry, modulate the properties of our polymers in order to generate complex behaviors. And since we are here about a workshop on controlled delivery, I will try to focus on this topic through three main examples. The control of drug delivery through self-assembling structures, the modulation of the biological activity of polymers through modification, and drug-eluting medical devices. So to start with the first topic, what I'd like to show you is that we can control drug delivery with simple parameters, simple synthetic parameters. I will more concentrate on the materials here, since I am a polymer chemist. And I'd like to show you those simple parameters with, as the first one, the amphiphilicity. So this is something which is really known. most micelles which are used for drug delivery are diblock micelles, mainly based on PEG hydrophilic segments and PLA hydrophobic ones. Once they are put into solution, you can form micelles, and most of them are spherical micelles. But it's also possible, just by playing with the ratios between hydrophilic and hydrophobic units, to end up with more strange or exotic structures like vesicles, phylomicelles, which are also called worm micelles. And what is interesting with those structures, which are anisotropic structures, is that it has been demonstrated that they can circulate much longer in bloodstream. For example, 10 times higher circulation for the anisotropic micelles compared to the spherical ones. So we were interested in the group, for example, to modulate the pharmacokinetics of the delivery system by changing the morphology of the diblock copolymers resulting assemblies. And that has been done with very highly hydrophilic systems. You've got the ratio of ethylene oxide units here. For the two extreme values, we have spherical micelles. But then if we go in between at 4.5, we obtain that kind of anisotropic micelles. And what is interesting is that you don't have the driving force of this self-assembly is not the architecture of your polymer, because it's obtained with diblock and triblock copolymers. This is more the ratio. If you keep the ratio constant, then you will end up with such assemblies. This is the first example of how to modulate through the amphiphilicity the morphology of your systems. Another thing we tried to do was to modulate the nature of the polymer segments. As I said, most systems are based on PLA and PEG, but we can switch the hydrophilic segments for polyesters, which will be obtained through the polymerization of hydrophilic lactones. You've got here lactones with carboxylic protected groups. And we can do exactly the same for the lactide. Instead of using exyl lactide, we can use diexyl or monoexyl lactide. So we will increase the hydrophobicity of the core of the micelles. So about some results about those systems, we prepared various diblock copolymers. You've got here an example where we kept constant the hydrophobic segment, and we just varied the nature of the hydrophilic segment. And you can see that there is a strong influence in terms of size of the micelles. The macromolecular weight was kept constant for all systems, and when you're using an ionic shell, you've got repulsions, electrostatic repulsion in the shell, so you've got a larger micelle compared to the non-ionic classical system with PEG. That's the first thing that may influence the circulation of your micelles. Another thing which is influenced by changing the segments is if you change the core nature. You've got here the PLA micelles, and then we've got the monoexyl PLA, diexyl PLA, and you can see that stability of the micelles over time is really increased when you change the hydrophobicity of the core of your micelles. And beside this, you can also entrap much more hydrophobic drug in these micelles compared to the PLA micelles. So that's another way to play around with micelles. Now I'd like to switch to the second parameter I'd like to focus on, which is the crystallinity of your systems. You may all know that PLA can be obtained under semi-crystalline phase, and you've got two kinds of monomers, D-lactide, L-lactide. You can have the homopolymers, which are corresponding to those monomers, and they are semi-crystalline thanks to the formation of helices in the structures, in the crystalline structure. If you do a mixture of both these homopolymers, you will end up with a much more closer interpretation of the crystalline phases, which is called stereocomplexation. And stereocomplexation will induce a higher stability for your system and higher crystallinity. This can be used, for example, for the formation of more stable micelles again, but also for gels. So that's what we've done here. You've got in those syringes triblock copolymers, PLLA, PEG, PLLA, or PDLA, PEG, PDLA. You've got injectable solutions without any co-solvent, and when you mix together the solutions, thanks to this stereocomplexation, you will start to build up a gel, which is shown here. And this gel is stronger in structure than the gel obtained just by normal crystallization without stereocomplexation. And you can see here the effect in terms of release for a model protein, which is BSA in that case. What is interesting here is the burst, which is really decreased when you use a more stereocomplex gel. That's a way of modulating, again, the burst release, which is not really something that you want to have. Last parameter about those self-assembling structures is the use of electrostatic interactions between the polymer and the drug. This is something which is known for coatings, for example, but much less for self-assembling structures. For example, you've got all the polyacrylic compounds in pharmacy which are used for that kind of interactive coatings. Here, in that case, we're interested to provide other kinds of self-assemblies. We are not talking anymore about micelles, but about aggregates. If you use random amphiphilic copolymers, you can form in water, depending on the composition, aggregates, and you can have either intra-nanoaggregates, which are independent on the dissolution and concentration, and you can also have inter-nanoaggregates if you have linear structures. So it's just depending on the ratios of hydrophobic moieties in your polymer and the structures of the monomers. In all cases, what is interesting is that you form nano-hydrophobic domains in those nanoaggregates, which can be used for the entrapment of LPI. So we've done this first as a proof of concept with biostable structures. We've got here a biostable backbone with alkyl pending chains and carboxylic groups. So we have a random polyanionic copolymer, which can be used to interact with clofazimine. Clofazimine is a drug which is highly hydrophobic, poorly soluble, but which has a cationic character. So when you associate both those structures, you will have a synergy between the hydrophobic association and electrostatic association, so that you can increase the solubility by a factor of 4,500 in that case, compared to pure clofazimine here in water in PBS. So this means there is a strong synergy, and we wanted to apply this strategy to degradable systems again. So here is a little bit of the chemistry we are doing. I won't go into details, but we started with this compound, and in 5 steps we generated a family of lactones, which are derivatives of the dimethyl malic compound. We've got an ester group here, which can be changed depending on what we want to obtain. And by copolymerization of those monomers, we can generate a family of polydimethyl malic acid derivatives, which are degradable, polyanionic, and random copolymers. Again, in terms of formation of self-assembling, those structures, because we have long alkyl chain here on the backbone, they will form more intra-nanoaggregates. We don't have any critical aggregation concentration. And it's possible to use those systems for the solubilization of drugs again. I just show you the effect here of the pH on the solubilization. If you use pyrene just as a model compound, it's a neutral one. It's not depending on the pH, so you have a solubilization of the pyrene, which is constant more or less over the pH range. If you go now to the methylamine pyrene, with a pK of 9, you always have your structure which is protonated, which means you always have the interaction with your polymer. And in that case, you have a very, very high solubilization. I don't have the values for the pure compound without polymer, but it's less, very less compared to the polymer. For the clofazimine again, we can see that because of the pKa, which is around 6.6, if we have a protonated clofazimine, we have a high incorporation, which is decreasing with pH. So that's one of the examples about these electrostatic interactions. Last, since we had those polyanionic copolyesters, we wanted also to have dependent systems with cationic groups. We generated these lactones, which are valeriolactone with an amino group protected here. And by copolymerization of these monomers with caprolactone, we were able to generate the similar structures, polycationic degradable random copolymers. But in that case, hydrophobicity is linearly included into the backbone, which means you will form the inter-nanoaggregates. They have a CAC, which is about 0.05% weight in our case. And you generate a mixture of 2 structures, small aggregates, about 50 nanometers, and larger ones, which are more like flower-like micelles, something like this, or aggregates, of about 250 nanometers. Since the structures are cationic, we had to be sure that they were not toxic, because polycations are known to be cytotoxic and hemotoxic mainly. I just show you cytocompatibility. Hemocompatibility is described here. But what we've shown is that compared to the polylysine, because we don't have present the amino group exactly at the same position compared to polylysine, we have a very decreased cytotoxicity. Here in red, you've got the cytotoxicity after 7 days of the proliferation, not cytotoxicity, but proliferation of fibroblasts in presence of polylysine after 7 days. Here you've got the control. And here you've got the homopolymers in light green and copolymers in dark green. And you can see that we really improve the compatibility as soon as we mix the polyamino variolactone with a little bit of caprolactone. So we have systems which are able to be used, that can be used for at least cell-contacting applications. And we showed also that it's possible to encapsulate drugs, but in that case, hydrophobic, non-soluble drugs with an anionic character. I won't go into details again. I switch now to the second part of my talk, which is more dealing about the modulation activity of polymers by, in that case, not modification of the polyesters themselves. A first application I'd like to show you is about the synthesis of antibacterial surfaces. Tomorrow we'll have some more details in the presentation of Professor McNeil. But one of the main problems when you work with medical devices and implants is the infection which appears in most cases just after surgery. You've got bacteria which are present at the site of surgery, and they will eventually proliferate and form biofilm on the surface of your material. Once they form this biofilm, they are not anymore sensitive to antibiotics, and that's one of the main problems with 50% of all nosocomial disease which are, or infections which are associated with such biofilms. So, as polymer chemists, we wanted to find a solution with polymers, and we chose to work with polyquaternary ammoniums because those polymers are active against bacteria in a non-specific way. It's just a destabilization of the bacterial membrane. We don't have other mechanisms, and it means it's working with a broad spectrum of bacteria. Another thing we wanted to do is to covalently link these antimicrobial or antibacterial polyquaternary ammoniums onto surfaces of polyesters in order to have a permanent activity and a non-leaching surface. So to do so, we had to prepare both the polymers, active polymers, and the modified or functionalizable surface. For the surface, we came back to our basics, more or less in 2000. The group published on the anionic activation of polyesters. You can see here that we started with a PCl, and by activation with a strong base, we can generate this macro polycarbanionic structure. And this intermediate can be used either to produce, by reaction with small molecules, functionalized PCLs, or we can also use this intermediate as a macro initiator to produce polymers which are graft PCL with hydrophilic side chains, for example, polylysine. So this strategy was quite versatile, and we wanted to use it on surfaces. So we started from PLA, processed the PLA under the shape of plates in that case. We had to modify slightly the conditions of anionic activations in order to avoid any hydrolysis, because PLA is more sensitive than the PCl towards hydrolysis. And after reaction with propargyl bromide, we ended up with this kind of clickable PLA surfaces. What is interesting with those surfaces is that they are shelf-ready, stable, so that we can functionalize the surface as a function of the need, for example, antibacterial need in that case. Coming back to these polyquaternary ammoniums, the antibacterial active agent, we chose to work with dimethylaminoethyl methacrylate as a monomer. We obtained well-defined polymers by ATRP polymerization because we wanted to control the molecular weight and study the influence of the molecular weight on the activity. You can see here that the initiator we used for the polymerization was introduced as a chain N with an azido group for the click chemistry. We checked that the azido group was remaining after chemistry by various techniques. And after quaternization of the structure, we realized the click chemistry between the polyquaternary ammoniums and the surfaces in aqueous media. You can see here one of the characterizations we carried out, which is XPS. You can see here for the neat PLA, without modification, there is no nitrogen appearing, no peak corresponding to nitrogen, whereas on the modified PLA, you've got this peak corresponding to nitrogen of the polyquaternary ammoniums. So that was the chemistry part. Then, of course, we're interested in evaluating our systems, both in terms of adhesion of the bacteria on the surface and biofilm formation. First, for the adhesion, what we did, we prepared a whole family of modified plates. As I said, thanks to ATRP, we were able to generate control molecular weights for polyquaternary ammoniums. The alkylating agent used for the quaternization was chosen between methyl or heptyl group to see the influence of this group, because it's known that there is a strong influence. And following this, we put in contact the plates with various bacterial strains. We incubated and counted the number of adherent bacteria. You've got here the results for one strain, Escherichia coli, which is gram-negative, and you've got the colony-forming units here. You've got the pure PLA without modification. You can see that we have a fully covered structure and surface, and as soon as you start to modify your surface with the polyquaternary ammoniums, you decrease the number of adherent bacteria, and the longer the polymer, the better the effect in that case. Same thing, here you've got the heptyl group, which is much more efficient in terms of anti-adherence activity in that case. So we are quite happy with this. We also visualized these results with green fluorescent expressing Escherichia coli. We've got here the PLA, which is fully covered, and the best modified plate, which is not covered at all, at least with visualization. We also did this with other bacteria. We wanted to be sure that most of the bacteria responsible for nosocomial infection were sensitive to the structures, to the modified surfaces. You can see here 4 types of bacteria. Within that also, this is not only anti-adherence, but it's due to the bactericidal effect of the polyquaternary ammonium, which has been visualized through a live-ended assay. You've got non-modified plates here, where the bacteria are alive. You've got dead bacteria on the modified plates. And finally, we just recently worked on the biofilm formation of these systems. And under drastic conditions, what you can see here, we've got the control. You've got 2 kinds of bacteria, and for both types of bacteria, we've got a decrease of 60% of the biofilm, which is quite encouraging. It's not perfect, but we'll still continue to work on this in this strategy. So that's what I wanted to show you about modification of surfaces for one particular topic, which is antibacterial activity. If now I come to another example, another problem if you work with medical devices made of polymers is that they are non-visible, especially by MRI techniques. So surgeons asked us if it was possible maybe to make the polymers visible, and the reason is because MRI is the most widely used non-invasive technique in hospitals. The problem is that if you go to hospitals for MRI visualization, you will be injected a solution which is highly concentrated in a contrast agent, and the active species here is the gadolinium ion. This gadolinium ion is quite toxic. That's why it's embedded in this ligand structure. It's complex. And the gadolinium will influence the reaction time of the water surrounding the contrast agent, and that gives rise to the signal. The problem is that all these systems are water-soluble systems which cannot be entrapped into polymers, which are the base of our medical devices. If you try to do a mixture of such contrast agent into, for example, PLA, you will have a release, an immediate release, which means that you will lose the signal after quite a short period of time. So we wanted to obtain polymers that were hydrophobic to stay in place on the prosthetic materials. And the aim, at least for the surgeons, is to evaluate the post-fixation of the prosthesis, is to visualize the tissular integration, and eventually, if the polymer is degradable, to see the medical fate, the fate of the medical device. So the solution we proposed was to, again, synthesize new monomers You've got here a propagelated caprolactam, which was copolymerized with caprolactam to generate this family of copolymers. We have PCLs with control ratios of propagelated units. In parallel to this, we prepared this contrast agent with azido groups again for click chemistry. And by combining those two structures in click reaction, we obtain this kind of macromolecular contrast agent. Of course, since gadolinium is quite toxic, as I said, we wanted to check the stability of the complex in physiologically mimicking systems. This was part of the training period of one of the students from Palermo, Barbara Porcio. And what she found is you've got here in green the PBS control. We used ICP-MS as a technique to visualize the release of gadolinium. PBS control is giving you this baseline. Green dots are PCL films without any gadolinium inside. You can see it's always around the PBS control. And the films formed from our polymers give exactly the same thing. So it means we don't have any release. And this is a scale, log scale in that case. So we have stable complex on the polymer, which means we will not release the gadolinium. In terms of effect now, you can see here we've got the effect of gadolinium ratio, which is immobilized on the PCL, on the relaxation time of the PCL protons. The ratios are quite low, below 3.5. You can see that as soon as you have modification of your polyester, you have a strong effect in terms of decrease of the relaxation time. And what is interesting is that above 2.5% of gadolinium on your structure, you already have a plateau. So you don't need to have a large amount of gadolinium into your polymer. In terms of in vitro visualization now, you can see here MRI visualization of PCL, which is not visible. And then with increasing ratios of gadolinium immobilized on the PCL, you can see here the signal appearing with an optimum which is around 2.6. If you go higher, again, you have a decrease of the signal. So this, this was quite interesting for us, and we wanted to transfer this in vivo. And we've done more or less the same approach with another polymer, which was coated onto prosthetic meshes. Those prosthetic meshes are the one used for hernia repair or reinforcement. They are also used by gynecologists. And what we did, we did the coating of this polymer onto the biostable polypropylene meshes. Then we implanted them into rats subcutaneously and intramuscularly. And you can see here on two planes the meshes, which appear here on the sagittal plane and here on the coronal one. We can do a 3D reconstruction of this system to have more information on the localization of the meshes, and that's the tricky part, always the film. Normally we see here, that's a 3D reconstruction of the meshes implanted in the rats. You've got the two meshes which are present. You can see the porosity and localization of the two meshes, and that was the first example of MRI visible polymers, which are in that case degradable coatings, but hydrophobic. It means with the PCR we have a very slow degradation, and we'll keep this signal quite a long time. So with this, I will come to the last part of my talk. I'm coming back to more drug delivery and control release, but in that case on scaffolds for tissue engineering and also on prosthesis, so more classical things. Coming back to antibacterial activity, we were interested again with the surgeons to provide a coating that may be able to liberate, to release two kinds of antibiotics in order to avoid any emergence of resistance and the following nosocomial problems that appear. What is available in the clinic right now, when you are you have a surgery which is done, is to have an injection of ofloxacin and augmentin, which is the French name, but this is a mixture of amoxicillin and clavulanic acid. When you use this system entrapped in coatings, the problem is that mainly you have an instability of your coating towards sterilization. You will lose the activity of your of your drugs. So we decided to switch to another system, which is ofloxacin and rifampicin. We wanted to have both types of bacteria, gram-negative and gram-positive, which could be affected. And we prepared some coatings by spray coating technique around commercially available meshes. Those meshes are both biostable again. Polypropylene threads are used to prepare those meshes. And you've got here the coatings, one coating containing the drug and the reservoir, another one which is just here the polymer without any drug inside, just to modulate the drug release profile. If you use this bilayer strategy, that's what you obtain, a burst effect, massive burst effect. You don't have perennity of your activity through the time. But then if you use a 3-layer coating, we played around quite a long time to find the proper conditions, proper external layer to slow down the release, etc. But if you do so, 3 layers, one with only ofloxacin at the inner layer, intermediate layer with rifampicin, and PCl-PLA mixture at the outer layer, you've got a much more controlled release in that case. And what is important to see here is that we have a release which is almost quantitative after 100 hours. In terms of morphology, if you do a coating onto your systems, of course, you don't want to change the mechanical properties of the implants. And that's what we checked. The coatings are quite, are not really thick. You've got about 20 micron as a thickness. We still can see the macroporous structure of the native meshes, so we can implant them. And we had no delamination between the layers, which is important if you want to implant your system. In terms of activity now, again, anti-adherence and anti-biofilm activity. You can see here that we have no adherence after 72 hours with Escherichia coli and S. epidermidis. We have a decrease by 80% of the biofilm formation. And what is interesting is that we have an inhibition diameter against, again the 2 bacterial strain, which is active up to 72 hours. And the main problem when you have the surgery are the first critical hours. If you start to have infection in the first hours, then it may be a major problem. If you still have this inhibition after 72 hours, you really decrease the risk. Well, that's what we wanted to obtain in that case with those releasing coatings. And now I come to the last example I'd like to show you today. This work was done mainly in Geneva, where I spent some time. In collaboration also with somebody from Bari, Delia Mandrakia, that some of you may know. We were interested in working in that case with nanofibers. Tomorrow again, I think there is some electrospinning stuff that will be discussed. We wanted to work on vascular grafts. It was in 2006, so there were not so many things done on electrospinning. And the aim was to provide small diameter vascular grafts. Because if you use the normal synthetic prosthesis, they are available, they are efficient, they are made of biostable PTFA or Dacron. But the problem is that if you go below the diameter of 6 millimeters, then you have some specific problems like aneurysm and mostly hyperplasia, which is an uncontrolled development of the endothelial layers in the inner part of your graft. And you want to avoid this in order to avoid the blocking of your graft. So we had to provide a degradable structure. We wanted this structure to mimic the natural vessel properties and eventually morphologies. And we prepared those grafts by electrospinning of PCl solutions. You can see here the grafts that were obtained, so small vascular grafts, 2 millimeter as an inner diameter. And we worked on the process of the spinning, the various parameters, to obtain morphologies which were controlled, remove all the defects like the beads here. And we did this optimization more in terms of mechanical properties. You've got here an example of optimization. We look at the stress at break that can bear the graft. If we use a non-optimized graft, here the red dot on the left corner, you've got after implantation an aneurysm that appears, so the rat is dead. But then, of course, if you optimize properly your mechanical properties, you have this kind of angiography. You can see here the prosthesis is between the 2 arrows, the white arrows, and we don't see anything. There is no problem of aneurysm after implantation. So having this in hand, we wanted, of course, to evaluate this in vivo. What you want to check in vivo when you implant such systems is that you have 3 phases. That's more or less the basic for tissue engineering. You want to see in the first stage the recruitment of macrophages from the immune system. Then you want to see appearing a recruitment of more specific cells like endothelial cells in the inner part of your graft, small muscle cells in the graft itself. You should be able, if you have a degradable polymer, to see also degradation of the polymer during this stage. And finally, what you should obtain is a regenerated tissue. So we looked at this by implantation in rats on the abdominal aorta of rats. And you can see here the graft in the aorta. And after 24 weeks, you've got here an example of histological cut. And what you've got here is the luminal part, the internal part of the graft. That's the wall of the graft. And you can see appearing neovascularization to provide nutriments, macrophages as expected, and also endothelialization, which comes as soon as 3 weeks after implantation. You've got full coverage of those systems. The problem is that you don't have a monolayer of endothelial cells. You have only a multi-layer, and that's what you want to avoid because it's more or less already hyperplasia in that case. So what was done, especially by Delia Mandrakia, was to entrap some paclitaxel, an anti-proliferative drug, into those nanofibers. We looked at the release of drug depending on the concentration over time. And then we looked at the effect of those eluting nanofibers into the meshes, into the grafts. You can see again, without drug, this multi-layer. And when we use the paclitaxel eluting systems, we have a nice monolayer which is obtained after 12 weeks, I think is here. Well, that's the main result we obtain with those structures. People in Geneva still work on these systems. There are some other problems that arise in those systems, so it's not perfect. But that's what I wanted to show you about how to process maybe the degradable polymers in order to go to tissue engineering. So with this, I'd like to conclude. What I wanted to show you is that by simple chemistry, we can obtain monomers, polymers, modify the polymers in order to fit into the various applications, biomedical applications that may require those degradability. And we can also play nicely with the polymer shapes, again, to really get closer from application. So with this, I'd like to thank, of course, all the people involved in those various projects, especially Professor Kudan, which is the head of the department. I'd like to thank the organization committee and especially Professor Caballero for inviting me, and I thank you for your kind attention. Thank you.
Thank you for your impressive and huge presentation with a lot of data. And there are now questions.
That was absolutely excellent. A bit difficult to know where to begin, so I'll just confine myself to the antibacterial surfaces. I'm not a chemist. I could follow what you're saying, but I'm not sure I know the answer to this one. Could you use that approach to put that antibacterial surface onto a range of materials, like urinary catheters, like endoscopes?
As long as there are polyesters, yes. Because this strategy was developed for polyesters modification. So if we have, like, for example, interferences screws or plates for osteoconduction or whatever, we can modify the plates. But that's more for the PLA and PCL, PLGA eventually. So polyesters, basically.
Okay. Yeah. And second question, then I'll hand it over. People always say that you can always develop an antibacterial surface which works for a day or two, but then the bacteria come back. Mm-hmm. Now, you made the point that the big problem is immediately post-surgery, which I agree. But have you studied bacteria on the surfaces for several days?
So we are now up to 72 hours. And we have a small problem because then we decrease the activity because dead bacteria, under the static conditions we're using for the test, because the problem is to find a proper test, under the static in vitro conditions which are used, the dead bacteria starts to form a bed of dead bacteria, and then we have a passivation of the surface, and we need to wash away the surface to have again the activity. And we are trying to find a way to in vitro evaluate maybe the cleaning of the surface by physiological fluids or something like this. But to date, we did not find any test in that way. That's a major problem. The test, I think, is also kind of brings a bias.
Okay. Thank you.
Sorry.
Really, thanks for the fantastic presentation. In the case of a system with the modified surface, which kind of technique did you use to quantify the degree, the derivatization of the system?
On the surface itself?
Yes, yeah.
So what we used was only XPS. So we have an average, average composition of the surface, which corresponds to 10% of the surface, which is covered by polyquaternary ammoniums. But it's not the real answer, and we don't have density of grafting, for example, to date.
Other questions?
More than questions, I have a couple of curiosities. But first, I would like to thank you for your interest in electrons.
Welcome.
The curiosities are both referred to the first part of your presentation, the drug delivery and assembly. A couple of times you said these systems are more stable. Well, which kind of stability are you referring to? Physical, chemical, or both? And the second point is about the route of administration. Drug delivery will be oral or, in principle, also injectable?
So normally we would like to have injectable systems to answer your second question. Of course, depending on the structure, for example, for the cationic structures, we can also use subcutaneous injection because we will have interaction with the tissues, and that's something that may be okay because of the cationic character. Even if we don't have hemoaggregation and cytocompatibility, we maybe would prefer not to inject those systems, except if they are complex, for example, with DNA for transfection or things like that. But mainly for the neutral systems, more injection. For the other ones, IV injection. For the other ones, subcutaneous injection.
Thank you.
And
Other
for the
questions?
first question about stability, it's mainly the physical stability, but which is also driven by the chemical stability. For example, for the hydrophobic core, hydrophobization of the core, that brings, thanks to a decrease of the sensitivity of the polymers towards hydrolysis, it brings the physical stability. So it's a mixture of the two.
Thank you.
If no other questions, I have just... okay.
Which ones?
I know, but in general, I know you show a lot of polymers.
We did DSCs with those polymers, and for example, the cationic ones show a strong fusion peak.
Okay, what about the TG transition?
TG, for those ones, I'm sorry, I will have to go back to the data, but we don't have always TG. Well, at least we don't see it. It doesn't mean we don't have one.
Okay.
For the cationic ones, I have to check. Really, I'm sorry, I would not say.
Okay, thank you.
You're welcome.
Any other question?
Just a curiosity, because of course you have spoken about a lot of polymers. Of course, I think you modulate the biodegradation time.
Yeah, depending, of course, the more. For example, for the carboxylic or cationic compounds, or amino-acid compounds, we have degradation which is much faster than the neutral ones. For example, it was demonstrated with carboxylated PCl, which is degraded about 6 times faster than the normal PCl. And with the amino groups, you can have those aminolysis, so you increase again the degradation. So all the polymers are different in terms of degradation as a result of their structure.
Okay, thank you.
Okay, thank you again, Professor Nottelet.